You're listening to The Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hello, Interactome listeners. It's Maia here to explain RNA. Woo! Very exciting. So this is part 2 out of 3 of a series of podcast episodes in which we'll be going through each part of the central dogma. So if you haven't checked it out already, you should check out our episode about genes, DNA, and genetics. But today we'll be talking about RNA. So I am looking forward to discussing all of the wonderful things that pertain to this amazing nucleic acid. And in the future, we'll also have an episode about proteins, and that'll tie up together this little mini-series. So,
proteins!
Woo! Exciting! But first, let's hear who else is on the pod today. And if you could introduce yourself and tell me what emotions, what feelings you feel when I say RNA to you.
I honestly love that question. My name's Joe. I go.
Wait.
That's like a bit that I've done like 3 times. Listeners who are recurring are probably like, Natalie, please let it go. No, I'm Natalie. I'm 1/6 of the Interactome. And when I think about RNA, so this is going to be cheesy, and we're going to get into this in a little bit. But right now, I think hope. Right? Because of the Nobel Prize, because of what RNA was able to accomplish, or the mRNA vaccine was able to accomplish as part of the COVID pandemic. And I also think of innovation. So hope and innovation are the 2 that come to mind.
Wow, amazing.
Hey everyone, I'm the real Joe. When I hear RNA, I feel a lot of excitement because similar to what Natalie mentioned, there's been a lot of really cool things going on with RNA, and there's going to be a lot of even like really cool things going on with RNA in the future. I also, like, a bunch of my research has kind of delved into some of the nitty gritty weird aspects of RNA that people just aren't really paying attention to. So we'll get into that. But yeah, it's just like RNA is really cool for many different reasons. And yeah, maybe I won't spoil it any further.
And I guess I'll wrap up intros by saying, I guess I never said what feelings RNA elicits in me.
Yeah, Maia.
But I don't know. I feel like my answer is weird now because I settled on 2 words for my emotions about RNA. One is nostalgia, and the other is fear. Fear.
Those are 2 great descriptive words.
Wow.
I will say, yeah, I do agree that there's a lot of very exciting things happening in the RNA field, and we'll get to that a little later. But for me, it's nostalgia because RNA is what inspired me to do a career in research. So when I was learning about it in biology classes, I was like, oh, that's a neat little molecule. I want to learn more. So I'll say a little bit more about this later in the episode, but it's kind of like what got me hooked into a career as a research scientist. And also fear, because maybe, Joe, you can relate to this somehow, but RNA is like a little tricky to work with in the lab.
Oh, it is.
So when I was working with RNA in my old lab, my PI or like my supervisor would like always scare me. She'd be like, has to be on ice, like don't leave it out for too long, like work as quickly as possible, like don't let it like degrade, and it was so scary. So I was always so afraid to work with it because we'll get into it, but RNA is very unstable. So I am afraid when I hear it. It stresses me out. But also, there are a number of other very cool things that are unrelated to that as well.
I have a story related to that too. I'd love to get into it a little farther.
Okay. Cool. But I guess we should kind of define what exactly RNA is. So RNA stands for ribonucleic acid, so that's RNA, ribonucleic acid. And it has 3 components that make it up. So there's the ribose sugar, which is one of the parts. So that's the R that's contributing to the name. And in addition to the ribose sugar, there's a second part called a base. So these are different molecules that are very important for kind of encoding information that gets passed around in the cell. There's 4 different kind of bases, and they are adenine, uracil, guanine, and cytosine. If you listen to our genes and genetics and DNA episode, you'll remember that DNA also has bases. And in RNA, the bases are the same except for uracil, which is unique to RNA. And the last part of RNA, part number 3, is a phosphate. So that makes up the little backbone of this whole RNA molecule and is kind of what allows for all of these separate little RNA molecules to be connected together so that they can make long, short chains, et cetera, et cetera. I guess kind of the key things that are different when you're thinking about RNA versus DNA is that, for one, I mentioned that RNA has that uracil group, which is unique to RNA, but RNA also has this reactive hydroxyl group on the ribose sugar, and DNA does not. So that's why DNA is called deoxyribonucleic acid. No oxy, deoxy. Wow. So those are kind of the bigger differences between our 2 favorite nucleic acids that are essential building blocks for life.
Yeah, and I think we can talk a little bit about too, what that actual kind of deoxy means. Before this episode too, I was chatting with my boyfriend, and he was like, "Oh yeah, what does the R stand for in RNA?" And I was saying ribose and deoxy, and the reason that and I believe, and Joe and Maia, correct me if I'm wrong, but one of the reasons it's so tricky to work with is because of that oxygen on the ribose that makes it much more reactive, less stable. Oxygen is an element that takes electrons away from other elements. It's a really, I learned it as a really greedy element. And so it takes all the electrons, and that has ripple effects on the other molecular structures around it.
Yes, you're exactly right. So that oxygen in the hydroxyl group on the RNA was the reason for my fear when I think about RNA, because it does make it quite unstable.
And hey, I just guessed on all of that. I was like, I think this is right, so I'm pretty proud of that one.
It's very reactive, and it's to get into it a little more, having that hydroxyl or OH oxygen hydrogen group on a sugar just makes it a lot easier for other enzymes and stuff like that to kind of attack the RNA and then just kind of break up the molecule. So it's very tragic, but it happens.
Yeah. I think a little just to kind of put this into reference for our listeners. It's we talk about this molecule being unstable, but if it was so unstable that it couldn't really do anything,
so true.
You wouldn't have a functional cell. So it is we'll get into a little bit, but RNA has a lot of different functions, and oftentimes those functions are relatively the time for which RNA has to perform a function is relatively short in comparison to DNA. DNA is it's as one of my former lab mentors said, DNA is the world's most stable molecule. It can last for a long time. I don't know if that's actually it actually is the world's most stable molecule, but you can take DNA and just leave it in a tube with some water just for years, and it will still be there. We literally can take DNA from woolly mammoths and sequence woolly mammoth DNA and stuff like that.
That's so
RNA cool. Is that's showing how stable DNA is. RNA is more easily degraded. It breaks down more easily, kind of as we're talking about. It's more easily attacked by enzymes, by proteins, by other like mechanisms going on in the cell. But it's still stable enough that it can get its job done in the time that it has to. So it's as what if DNA is kind of like the central like place in which the cell codes information, RNA is kind of like the middleman, or in its most like commonly well-known function. It carries messages from the nucleus, where all the DNA is, into the cytosol, where all the protein-making machinery is. And so, like, in that sense, you don't want your messages to last forever. Because if you're saying, oh, make protein A, but you don't actually need protein A, like, you don't actually need more protein A. If you're just constantly making tons and tons of protein A, and you keep sending instructions to make that, like, that's a problem. Like, cells have to be able to adapt. And so that lack of or reduced stability in comparison to DNA is actually very useful for RNA's function.
Yeah, and it's so interesting because I feel like cells have so many different ways of regulating RNAs as well. So not only just relating to their stability or their degradation, but even just like producing the mRNA there's so many layers and layers of regulation that undergoes that. So like, when do you want protein A? When should we make it? So I guess like to back it up a little, Joe mentioned that the kind of like most well-known function of RNA is to act as a middleman between DNA, which we know from our last podcast episode about genes and genetics, encodes information. Go listen. So the DNA encodes information for the proteins in your cells, which are doing kind of like a variety of functions, doing all the work in your cell to keep them happy and healthy. So RNAs are like the middleman, so they are produced from the DNA. So there's a specific cellular machine that produces the mRNA that has like a corresponding code that you can also find in the DNA. So if you remember those bases I talked about, the adenine, uracil, guanine, cytosine, when RNA is made, it also has those bases and just like kind of like in the same, it has like the same information that of the DNA that it is templating. So then since you have your RNA made from DNA, or rather to put it in a better way, your RNA using DNA as a template for the sequences that are found in the RNA. It has all these instructions, so these sequences that code for your protein, that another cellular machine will kind of like read and then produce the proteins based off of that code. So these are kind of like the big messenger RNAs that we talked about that are responsible for bringing these messages, like Joe mentioned, to become proteins. So what's part of a messenger RNA? We mentioned the code that's kind of like encoded into the messenger RNA. So there's sorry, let me back it up.
Can I ask a quick question?
Yeah. my train of thought just like fell off abruptly. I was like, ooh, messages, and I was like, what?
That happens to me all the time.
Yeah.
No, literally me all the time. Why is there a U in RNA and a T in DNA? What's the point of that?
I think it's a really good question. I'm going to say I haven't explicitly looked into this, but I think one of the functions is to distinguish between what is RNA and what is DNA, kind of. That's one mechanism. Also, I think this is a little, we're actually going to get into this later on, but RNA, like the uracil base as compared to the thymidine base, like there's actually some modifications that can be made to it that can indicate that, oh, I am a part of the cell. I am like supposed to be here, as compared to virus RNA, which doesn't have these modifications. So
that's so interesting.
there's probably a lot of other reasons why, but that's a few, I would say.
I think also uracil and thymine are pretty similar in structure as well. So like Joe was mentioning, we are probably going to talk about important uracil modification later on. But I think the difference between uracil and thymine is that thymine is methylated and uracil is not. So if I remember correctly, there's probably some kind of like process that ensures that like the thymines are methylated, and then they kind of like get put in the DNA. But evolutionarily wise, or like why, I'm not too sure. Joe, you look like you have an idea.
I just did a quick Google search, and honestly, it seems like a lot of the most commonly cited reasons are actually going back to that whole idea of stability, where like, because uracil is, as a structure, like uracil is kind of like actually like this like a 5 carbon ring of ring of 5 carbons and a ring of 6 carbons kind of stuck together, like that, like, and with a few little other atoms kind of coming off of it. For whatever reason, that structure is a little less stable than thymine, which is very similar structurally, but it has like a few different atoms in terms of its arrangement. And so this uracil is more easily able to like break down or degrade, which kind of goes like RNA doesn't really care, because I mean it's going to be broken down relatively quickly.
Scientists use Google too. That's one thing that we can learn from this episode.
Google's very important.
Also, yes, and note that Joe said the sources, plural. So if you're consulting Google, don't go with the first thing that pops up. Check out our source episode.
I think another important thing to note about RNA is that it has a really interesting structure. Like DNA is kind of like 2 rungs in a ladder that kind of like twists in a little helix shape. RNA is just like, imagine you had one strand of DNA sequence-wise. You could take that sequence, and instead of having another sequence that's entirely complementary to it, you could take this one strand and fold it in on itself in many different ways, and like twist and turn it, so you get all these wild shapes. That RNA can do that. So you can have many RNAs that are similar in their sequence, but given like that RNA can fold up into many different ways, something with a similar sequence may actually in 3D space be very different structurally. And so that's something that's very important when it comes to RNAs. The 3D structure of the RNA is very important for its function. And so a good example of that is transfer RNAs, or tRNAs. These are little guys that are actually kind of in a T shape. And
they're so cute.
They're so
adorable.
Cute. They're really cute. And their job is to actually, they're not like the normal messenger RNAs. They actually help read the messenger RNAs. And so their little job is to go and read every 3 bases and say for these 3 bases correspond to this kind of amino acid. So every tRNA is linked up to an amino acid, and the tRNA kind of like floats into the ribosome, the protein making machine. And as a RNA and a messenger RNA is run through the ribosome, kind of like a printer, this kind of like translates the information on the RNA into a protein. And we can get into that more in the protein episode, but that's another really cool function that that RNA can do. And I personally have a bias towards tRNAs. We'll get into that later, but just had to put that out there.
Yeah, for sure. And yeah, like you mentioned, RNA can adapt all of these interesting folds and structures, and they can do a lot of different functions, which is really interesting. And I feel like this leads into a kind of like debated but very well known and maybe is popular the right word? Maybe well known hypothesis in the RNA field. And that is the RNA World Hypothesis.
I was looking at Joe and I was about to make like a dun dun like noise too, but then Joe started making a noise. I did mine anyway, but
But yeah, the RNA World Hypothesis is kind of like controversial, depends on who you talk to. But I feel it's we'll see where we stand on this. But I would say it's pretty well known amongst biologists that are, I don't know, kind of interested in the origins of life. So kind of the big idea behind the RNA World Hypothesis is that, well, you know, in the beginning, when there was no life and everything was just primordial soup everywhere, life needed to find
chicken soup.
Yep, just Campbell's soup. When the universe was just soup, when Earth was just soup, there was no life. But
tasty.
If we bring ourselves back to those primordial times, you know, life needed to find a way to replicate itself. That's one of the big defining characteristics of what counts as a living thing. And scientists have hypothesized that RNA may be what's responsible for this. So they're thinking of RNA as a potential molecule that can self-replicate or auto-catalyze its own formation. And then maybe this is how all primordial cells kind of like went around and did their business, made other primordial cells maybe, before we had modern cells with like DNA and RNA and protein all together. But then since there's been some evidence, so Joe said that RNA can kind of like fold on itself, adopt all these structures. There's also other examples of RNAs in nature today that have catalytic properties. So there's rRNA, ribosomal RNA, which is a part of the ribosome, so the protein-making machine that we mentioned earlier. And the ribosomal RNA is embedded throughout the ribosome, so it's kind of like really, really in there. But what's cool about it is that it has a catalytic property in which it facilitates the interaction of the messenger RNA and the transfer RNA, so the mRNA and the tRNA. And it's kind of weird because why does it have catalytic properties if it's kind of just a nucleic acid? So the existence of these kind of catalytic RNAs have led scientists to postulate, oh, you know, since RNA can both contain genetic information, so have like the bases, the information that encodes for the amino acids, for the proteins, so it can have genetic information, but it can also have like protein-like properties where it catalyzes things. Maybe it was the kind of originator molecule way back when, years and years and years ago in evolutionary space.
I guess
how do we feel?
Yeah, I have a question. Joe, do you want to go first?
Yeah, I was just gonna say that maybe for our listeners we should discuss a little bit about what catalysis means.
I was gonna say that too, and a little bit of what the theory is behind RNA being able to do that when there was nothing, right? When Earth was soup, how did RNA then catalyze?
And I think therein lies the whole debate about
right.
About
okay.
the entire hypothesis. I think there's been a lot of interesting research that has come out like for or against this. I think it gets into more like philosophy rather than experimental science in a lot of cases, I think, which I'd love to get into maybe in another episode. But
yeah, I want to get your takes on it though, both of you. I mean, you both work with RNA. You're both, PhD candidates. I'd love to.
Yeah, I personally don't know enough yet about this specific aspect of RNA. It's a whole world out there, pun intended. But yeah.
I don't get the pun.
It's the RNA.
RNA World. RNA World.
It's
like we're the whole hypothesis, like the
RNA World Hypothesis.
Stuff, like the RNA World Hypothesis.
It's RNA's world. Yeah. We're just living in it.
Episode title.
Yeah, right there. Yeah, sorry, Maya. Do you have any other thoughts before we go on to catalysis?
Yeah, I can get into it a little more. I feel like I don't know. I feel like this is kind of like a polarizing topic, and I think you're right. It does kind of like wax on philosophical. I feel like there could be kind of like more informed kind of like evolutionary studies. Maybe I haven't looked deep enough, but I know people have kind of like simulated evolution of molecules kind of like from the start, like in their own labs, and there's some data that suggests pro RNA World Hypothesis there. But I also I'll tell you guys about like the arguments against the RNA world, but I think like a good context to remember this in is that like we're in soup. Like there's nothing like too complicated going on. I feel like the RNA we're thinking of right now is probably very different from what RNA was years, eons ago in the primordial soup.
That's true.
So I think
in our soup era.
In our soup era. And so I feel like it's probably like much less impressive like what RNA did in the past and like what cells were up to like years and years and years ago. So we'll keep that in mind.
Mm-hmm.
But yeah, but on to catalysis.
Yeah, so I would say you can the way I think about like the concept of catalysis is a catalyst or is something that facilitates a chemical reaction that wouldn't that is much less likely to happen without that thing kind of helping it along. So for example, if we have let's say we have a bunch of amino acids in that all the building blocks of proteins, or a bunch of actually even better, a bunch of RNA bases, like our ribonucleotides, all floating together in the primordial soup. If we were to kind of like wait around for all of them to like link up into a sequence, yeah, that might happen eventually, but honestly, like it might take billions and billions of years. But if we have a protein, or like a little machine that can actually like help it along, make it a little easier for that reaction to happen by bringing some of the building blocks closer together instead of having them float all over the place. That is what catalysis is. It decreases. Catalysis is something, or is the process of decreasing the activation energy of a chemical reaction. And that's what a lot of proteins do. Like they make something easier to happen. You have to put less energy into it. Like maybe it's because you have the molecules closer. Maybe it's because like you're pulling some electrons off of one molecule and putting it onto the other molecule. But I'd say that's the best way to, in my opinion, as I know it now, to think of catalysis. So RNA can do things like that. Like it can do things like a lot of proteins do, like all these weird enzymes that like go around and like cut DNA, for example. Like we could talk about that in an episode. CRISPR, I think some people have heard about it, where you kind of like this cool gene editing technology. That's an enzyme. That's catalyzing a reaction in which DNA is being cut. But that'd be hard to happen without that enzyme there. And RNA can do similar things too, which I think is really, really cool.
Yeah, and I think that gets into some of the arguments against the RNA world, interestingly. So I feel like the pro arguments kind of like rely on this like crux that, RNA can have catalytic like abilities, and that's why it could only it's feasible to think that maybe this is like the one molecule that cells needed to have in order to keep going and sustain life. But then there's also kind of like the counter argument, which you hinted at, Joe, where it's kind of like a chicken and egg thing. So for RNA, we mentioned that there are these long chains of bases that are kind of stuck together. So some people are like, well, how do you get the RNA without the enzyme in the first place? What came first? What is it? And people think that, maybe you need a really long, complicated RNA to be able to make the crazy shapes and do the cool catalytic functions. But then how do you get like the long RNA without something stitching it together? Maybe it just kind of happens over space and time, like a really long time. Maybe it's different from what we're thinking of. Go ahead. Yeah.
I just think it goes back to what you said about the RNA, that if, say this theory is true, the RNA that existed eons ago in this soup, we need to stop saying soup. It's not the RNA that's in our bodies now. It's not the RNA that's studied. So, I'm no scientist, but I just think that those points are kind of mute when you think of that.
Yeah, because I feel like there's a lot of, and that's like kind of like the fun things about theories is that like none of us can go time travel back into like the soup and like peer into the little cells and like see what's in there. That would be cool, and it would shut up the debate forever. But what's cool about like the RNA World Hypothesis, and I guess like this is kind of like very true in the spirit of science, is that it really depends on like the assumptions you're making, and that completely changes kind of like the conclusions you can draw. I think it's like kind of spicy and fun to have the RNA World Hypothesis. So it kind of like makes everybody kind of like reevaluate what they know, like what does make sense, what doesn't make sense given what we currently know. Is it safe to assume RNAs today are the same as RNAs years ago? That kind of thing. But I do have one more soup point that I want to bring up. Another popular counter argument for the RNA world is that you mentioned this a lot of times, and it's the reason why I'm afraid of RNA, is that it's very unstable. So like Joe was saying, it's not the end of the world if RNA is unstable, but if we think back to the soup, the primordial soup, prebiotic conditions are like kind of like, not like a nice 70-degree day where it's like sunny, it's like probably really different when the earth was first formed, right? And when life was like starting to take shape. So I was reading an article saying that, so like the primordial soup like is kind of harsh. Some people think that RNAs evolved on ice, where maybe it's more stable. And also, I guess, like, given the conditions of the Earth at that time, like, the overall acidity of the environment was, like, pretty acidic. So people, like, in the article, they called it, the primordial soup might not be a soup, but a vinaigrette instead. So, more food for thought, pun intended.
That was good.
Yeah, and acid isn't that great for RNA. It can break it down.
Yeah. RNA really had to go through a lot, whether or not it did come first.
That it did. And they're still getting broken down every day. Made and broken.
Yeah. But
every day.
An inspiration to us all. Yeah, but I thought I would share the name of that article because I think it kind of captures kind of like the spirit of debate around the RNA World Hypothesis. So the title of the article is called "The RNA World Hypothesis: The Worst Theory of the Early Evolution of Life, Except for All the Others." So yeah, good. I feel like there's probably a number of scientists that feel like have very strong opinions about this hypothesis, but there are some that are like, like it could be either or, and I think that was kind of like the spirit of this article, which kind of corroborated with a lot of other papers that I looked up, presented some cases for and against the RNA world. So I thought it was kind of funny.
And that's the beauty of science. There's always a debate about something.
Mm-hmm.
Oh, yeah.
Mm-hmm. So yeah, think about it, listeners. Are you for or against the RNA world? Some people think that there's a proteins first hypothesis where proteins actually came first before RNA, or maybe they co-evolved together. Much debate there, but we'll let you kind of stew on it, soup on it, and think about it.
Stew.
I did not know this episode was going to be about soup.
Well, you make the art for all the episodes, so I'm just imagining like you drawing like a cute little bowl of soup, like alphabet soup, and it just saying RNA inside.
I love it.
If that's not the cover art for this, everybody ignore me.
Thank you for giving me the idea. I love it. Also, I don't know. It's autumn. It's fall right now. I feel like a big cozy bowl of RNA soup. Is just what we need.
I mean, your soup probably does have some RNA in it.
Confirmed. There's definitely some in there.
I mean, if you got chicken, if you got veggies in there, like there's probably a little RNA.
So is
like,
all soup RNA soup?
I guess so.
I think
Nutrition.
I think that's the theory that we confirmed today.
Soup confirmed.
I mean, I think it'd be like, given the normal ways in which we make soup, I think it'd be very challenging not to have anything like just biological in there, because just like anything biological is going to have RNA. So,
it's like gluten-free, RNA-free soup.
Yeah, I think that's going to be a little harder than being gluten-free. But yeah. I think also another interesting thing, like Maia has made all of the art for all of our episodes as our extraordinary artist in residence.
Thank you.
I think that's a wonderful title for you. But she's also a full-time PhD student doing PhD things that have kind of, if I remember correctly, related to RNA.
Yeah.
So I'm very curious to hear.
Yeah, I can speak about it a little. I actually, so to be more accurate, my current PhD work has nothing to do with RNA, which is fun.
Was fake news.
No, it's cool. But I will always have a soft spot for RNA because it was what my first research experience was in. And the lab that I currently work in also specializes in RNA as well. I just happen to be doing a non-protein or non-RNA project. I just happen to be doing a non-RNA project because I'm built different. No, I'm kidding. But
Joe, have you not been listening? Maia's clearly afraid of RNA.
I'm afraid.
That's a good point.
My boss was like, I know. I was like, don't be afraid. I'll give you a different project. But yeah, maybe I can pop in on the protein episode and tell you guys what I'm up to there. But I still am tangentially around RNA. So I guess I told you guys that RNA is what inspired me to do a research career. So I learned about it in biology class. I was like, wow, that's neat. It seems like, I feel like I was an RNA World groupie, perhaps in undergrad. I was like, oh, so cool. It can do so many things that DNA can't. So I wanted to learn more. I emailed a bunch of RNA labs at my old university, looking for undergrad positions. So, I ended up working on a translation lab. So that's a process, by which the messenger RNAs, mRNAs, are produced into proteins. But in my journey, I also emailed a RNA lab. So there's so many different kinds of RNA with fun little acronyms. So I was sending emails to all of them. And I had a great time. Was afraid of working with RNA for a little bit, but learned a lot of really interesting biology. And in my undergraduate lab, we read a journal club paper. So we read a recently released study published from UC Berkeley about a really weird long RNA. And we talked about it in journal club, we discussed it, and then fast forward a few laters, I joined that lab that published that weird paper about the weird long RNAs. Woo! So it all comes full circle. And to get into those strange long RNAs, which is what my current lab studies, they're called LUTs. So that's the acronym, LUTs, very cute.
That's so cute.
It is really cute. So what it stands for is long undecoded transcript isoforms. And they're basically just longer RNAs compared to kind of like a normal RNA. So in our lab, we study, like, we're interested in looking at like different kind of fun, non-canonical, non-conventional methods of gene regulation. So how genes are expressed, that kind of thing, how they're expressed into RNA and into protein. So these LUTs, or long undecoded transcript isoforms, are one way of looking at that. And so we have like a specific set of genes that we were looking at, and each gene kind of has a corresponding mRNA, right? So we talked about that a little bit earlier. But the LUTI, some genes have a LUTI form of that RNA, where instead of like the normal length mRNA you might expect from the gene is made, a longer version is made. And this has interesting properties. So like, what are these like long boys doing in the cell? Who knows?
The long
and
boys.
we're still doing a lot of work to try to figure out the function of these LUTIs. But something interesting that they demonstrated was that they're able to control kind of the expression of the gene. So when you have the LUTIs, the long guys being made, it corresponds to a decrease in protein output from the same gene. So the thought is that these LUTIs are suppressive for protein production for those specific genes. And what was really cool was that my lab found that there's a specific protein, a transcription factor, that is responsible for kind of toggling the control of these normal mRNAs versus the longer guys, the LUTIs. So switching between the LUTIs and the mRNAs during distinct developmental stages. Yeah, so in my lab, we also study meiosis, so the process in which sex cells are made. And we found that there are a number of LUTIs that are temporally regulated, so turning on and off during distinct stages in meiosis, so throughout the developmental program. And this one transcription factor, which is called NDT80, it's a really important transcription factor for meiosis, regulating the expression of genes that help carry out meiosis. It was shown to be able to turn on these LUTIs, the long guys, and suppress the protein production of those genes of interests. So like Joe was saying, you might want to have like a protein or RNA at one time point, but not the other. That's kind of what this NDT80 transcription factor is doing, but kind of like through this weird way with long RNAs. Ta-da!
Very cool. Thank you for sharing. You've been on this podcast for so long, and I feel like this is the first time we're, hearing about the work you do. So
Behind
that's
the
so
scenes.
Cool.
Yeah.
And Joe, I think you also have an RNA story that you want to share with the class.
Yeah, absolutely. So, like Maia, I have historically been very scared of RNA because, when I was first working with my supervisor, that I was working with in college, doing research, he told me that humans actually just give off RNases, like these proteins that they're kind of like little Pac-Mans that just go around just chomping down on RNA and just breaking it up. And the reason they do that is to break down viruses that are made out of RNA or things along those lines. You don't want RNAs from other organisms kind of invading your body. That's just not good. And so the challenge is, as a person who is giving off a lot of RNases, who is trying to work with RNA, it's a little, you run the risk of having the RNAs break down on if you breathe on them wrong. So it can be challenging. And so my first project in the lab was to do RNA sequencing, where you basically your goal is to kind of see what the cell is thinking about. You basically pop it open, pull out all the RNAs, break out the RNAs into little pieces, and turn them into DNA so they're more stable and easy to sequence. You basically do DNA sequencing and kind of match the sequences you get up with the known human genome to be okay, we have a lot of this specific sequence in our sample, so it must mean that there's a lot of RNA from this region in the DNA that is being transcribed. For whatever reason, the cell is trying to read this specific sequence of DNA a lot and try and make proteins out of that. So I was doing that, and I spent a whole week doing the RNA sequencing protocol, just 8 hours at a time, just going through it. It was a long protocol. I was up very late. I was wow, I'm gonna do this first try. It's gonna be incredible. And I did my analysis to see how much RNA libraries I got, which is basically the DNA that you send for sequencing. after converting the RNA to DNA. And I had nothing. There was nothing in my sample at all.
Nice.
So like nothing at all. So a whole week of work was just lost, and it was very sad. And,
It'd be like that.
It really do.
To this day, I don't know like what exactly was happening there. Like maybe like it's possible that the reagents were faulty, but it was also my first time working with RNA, and I'd been warned. Oh yeah, just watch out. Like,
don't breathe. By the way,
there's a good chance that So
actually, can I posit what might have happened?
Yeah.
So one time in my undergrad lab where I worked with RNA and where I became afraid of it, one time we saw a spider on the bench, and then my PI was like, "That's got RNases on it. Get it." So I think a spider came and walked all over your samples, and then the RNases degraded it.
It's quite possible.
Yes.
But yeah, so I was kind of scared of working with RNA for a while. But then after college, I found myself doing cancer biology work, kind of trying to understand a protein that turned out like the way it was causing cells to die, like when we turned it on, was like in theory as a cancer treatment, like killing the cancer cells by turning on this protein just in the cancer cells. It was actually cutting up a very specific tRNA. And so this protein, Schlafen-12,
you're kidding,
was cutting up one specific tRNA. And basically, I was working alongside this incredible RNA biologist. Like, he was my mentor. He literally an absolute whiz with RNA. Like, like he lives and breathes RNA. It was so cool.
He breathes it, but he doesn't breathe on it.
Exactly. But like, I had a chance to learn from him like more about like how to work with RNA, and like I was very scared initially, but he was kind of just like, "Don't worry, Joe. Like, you got this. Like, it's okay. Like, if it goes wrong, like, it's all right. We can just use the previous like another sample, and it'll be fine." Like, and so I ended up purifying a bunch of RNA from cells, like to the point, like I got enough RNA that I could like see like this massive pellet at the bottom of my tube.
That's dope.
It was really cool. And like this white, like almost like pearl at the bottom of it. Like I started out with a large amount of cells. And I was looking at like all the RNA, not just the messenger RNA. And so I then ran it on a gel, and I had this beautiful, like, just like tons of different bands, but they were all like clear and crisp and clean. And he looked at me and he was like, "Joe, you have the hands for RNA." And I was like, "Oh my gosh." Like, it made me so happy. And
did it,
I think it's like, yeah. And obviously, like, the really interesting thing was he was like, you don't actually have to worry about it too much. Like, just wipe down the area, like, with your RNA killing, like, stuff. Just don't, like, sneeze on it. Like, don't worry too much, and it's probably going to be fine. And, like, RNA is a little less stable than you'd think. Or a little more stable than you might think. And so it turned out to be true. And it was just a really powerful experience. And now here I am. I'm doing a little different stuff right now. I'm working with trying to understand mitochondrial RNA. And because
go listen to our mitochondria episode if you haven't.
Mitochondria in your cells actually take in RNA from outside the cell, or from outside the mitochondria, and actually spit out RNA into the cytoplasm, of the cell that they're inside. And so there's this really interesting communication going on between the larger cell and the mitochondria, where they're sending RNAs back and forth from each other. And we're trying to learn more about that and how we can co-opt that for like sending RNAs to the mitochondria and back and forth. So yeah, it's a cool time.
Cool. And yeah, as you guys know, there's a lot of interesting RNA research that's going on, so many new developments, and I thought it would be fun if we close out the episode with a little discussion about current events.
Oh yeah.
So, as you may have known, maybe just a little bit, the 2023 Nobel Prize in Physiology and Medicine was awarded to RNA biologists Catalin Carrico and Drew Weissman for their work in the development of the COVID mRNA vaccine. Seem familiar? Maybe. So I think this was really cool because maybe just to my knowledge, it didn't seem like mRNA vaccines were a huge deal prior to the development of the COVID vaccine. So when you think about vaccines, what you might have heard is that usually people kind of like use a weakened or dead version of whatever disease-causing agent to build the vaccine. So when you get that weakened or dead version of like a virus or bug, your body was like, oh my God, an invader. And then it launches an immune response so that you can be protected if you encounter the real thing, if it happens. But since you're getting like a weakened or dead version of like the virus or germ or infectious agent, it's unlikely to do you much harm. So since then, for vaccine development, other than using those weakened or dead versions of viruses and germs, people have also traditionally used different kind of proteins isolated from the virus to help put into the vaccines. People have also used genetic material from viruses as a vaccine. And all of those have worked pretty effectively. I think we have probably received a lot of vaccines over our lifetime that uses one of these methods. But I think the main con for the development of these vaccines is that you need to use a lot of resources. You need to mobilize a lot of equipment and reagents to be able to produce the vaccine to a large scale. So for the weakened or dead versions of the virus, or the proteins from the virus, or the viral vector, genetic materials, like all of that goes into other types of vaccines, you need to large scale produce them in cell culture. However, I believe the idea for mRNA is that there's no need to use a cell culture system to grow all of the components that you need for the other types of vaccines. You can just synthesize mRNA in vitro, just like make a long little chain of mRNA. But from what I understand, the challenges before the COVID vaccine was that mRNA was hard to deliver and also induces an immune response once administered. But the idea is cool, right? So you could kind of synthesize an mRNA molecule, maybe for like a protein that the virus has, deliver it to the cells, deliver it to a person, and then they could launch their immune response and develop resistance to the disease-causing agent. So, yeah, that's kind of like the principle behind it. And Joe, I believe that you have some insight on kind of the Nobel Prize workings that happened for Dr. Carrico and Dr. Weissman.
Yeah, so actually, the Nobel Prize was awarded to Doctors Carrico and Weissman here at the University of Pennsylvania, which was very, very exciting. They have a lab here. So it was kind of a big deal. We all, like a bunch of people came and like celebrated them, and then we had a big toast. And I think they were getting a little overwhelmed by the amount of people who were there, but it's just a really exciting experience because, to be honest, the discovery that they made really like has saved a lot of lives, and it's really significantly like over like billions of people have received to my knowledge, that number may be incorrect, but to my knowledge, billions of people have received an mRNA vaccine, or at the least there have been billions of doses made. And so what they discovered is not exactly the way to, like, the idea of an mRNA vaccine. Like, that had been thought of before. It was that whenever you took your mRNA and just stuck it in a cell, like, it wouldn't always, like, you might get a little bit of the protein that you're trying to make, but like not enough where it could actually be like therapeutically useful. And part of the reason for that, Doctors Carrico and Weissman found out, was actually because of what we were talking about way earlier in the episode. mRNA, like that uracil that we were talking about, can actually like be slightly modified. Like that uracil base can be changed to a base called pseudouridine, which is very structurally similar but slightly different. And what that tells a cell is essentially, hey, I am part of you. I am not a virus. I am okay. Don't kill me. And so what they showed like a while ago actually was that if you if you take an mRNA, like if you make an mRNA and you change all your uracils to pseudouridine, the cell more easily accepts that, and it doesn't turn on the immune response as intensely as if you had a mRNA with just normal uracil. And so it turned out like initially people were not interested in this idea at all. It was to the point where Dr. Carrico, when she was trying to apply for funding, to get money for this research, couldn't actually get grants because people thought it wasn't necessary. Like maybe like this, I'm sure there are other things that went into this, but it's to a lot of people, they didn't think it was a worthwhile area to pursue, and that it was better to invest their money elsewhere. It got to the point where she actually ended up leaving the University of Pennsylvania and founding a biotech company because she couldn't, like she was kind of pushed out because she couldn't bring in funding. So this kind of illustrates one of the challenges that exists within academia. And certainly like we think greatly of this kind of work now, and it's clearly very important for a lot of people, but historically it was not really well appreciated or not considered to be super relevant. And so unfortunately Dr. Carrico, when she first started doing this work, was struggling to get funding, and she ended up bumping into this other person, Drew Weissman, at a copy machine, and they kind of had like a little
of
disagreement.
All places.
And they just started talking about mRNA and about like RNA modifications and her research, and they ended up having this wonderful partnership that blossomed into the ability to generally effectively, not just in principle, but effectively get proteins into a cell, or messages to make proteins into a cell, to give like a program to a cell to say, hey, make this protein, but also the basis for the mRNA vaccines. And so it's really incredible, but it was not a linear, not an easy journey. And there were a lot of people along the way who just weren't excited about this work, and as such, it was challenging to get funding initially. Now, certainly, there are many people making a lot of money off of this.
No question.
Seriously.
But it goes to show like how there are a lot of scientists out there whose work isn't always recognized, but at the drop of a hat, things can change and that work can be needed. And so I think it's a really powerful illustration. Sorry to rant for a long time, but I think it's a really, really cool story in many ways.
Yeah. And like I thought it was so cool how like the mRNA vaccine is like basically getting the cell to like do the work for you. So now that we have that we didn't do it, but like Doctors Carrico and Weissman kind of like figured out how to sneak the RNA past the cells and into them, so that they kind of like don't get destroyed by like the other cells thinking that it's like foreign RNA. Once the message is in there, cells can just make the COVID spike protein and then launch an immune response after it. So that's why I think like it's kind of crazy, because I feel like when all of like the funding and everything emergency funding was being poured into like developing the vaccine, once the mRNA vaccine was like hammered out, like it really took off. Like you were saying, Joe, like billions and billions of doses were made. People are able to manufacture it in a short time, probably because like you didn't need to like have like huge giant cell culture systems. You could just make RNA chemically, and it was a good thing to have when we all needed a vaccine very quickly for many people. So yeah.
Absolutely. And I think another thing is, this is not the end of RNA work. There's also a lot of work being done for RNA-based cancer vaccines. So like you can find like basically like code an RNA that like may make a protein that is like specific to a cancer rather than the rest of your cells. And so it's just a really cool field to like watch emerging right now. And there's so many things that will be happening down the road. So many different RNAs to play with, for sure.
100%. Yeah, and we didn't even touch on all of the different kinds of RNAs. There's like a lot of really interesting like new like sequencing technology that's out there. Lots of different therapeutics that are being made. So definitely is an exciting space to watch.
Absolutely.
Yeah. Well, I think that's about it. I think we could talk about RNA forever, but I think that could be maybe a part 2 episode. But yes, thank you so much for joining us, everyone.
Yeah,
Woo!
Thanks so much.
Thank you.
Follow us on Instagram, Twitter, LinkedIn. We're at Interactome Media.
I believe our Twitter handle is at the Interactome, and
at the Interactome, yeah.
Our Instagram handle is at the Interactome_media. And you can also find us at www.interactomedia.org.